Hi, thank you, Yuriko Desa. I'm a professor in the Department of Oceanography and Coastal Sciences. Let me start with my teaching activity. This spring semester, I'm teaching OCS 7175, Environmental Optics. In the fall semester, I'll be teaching advanced topics in remote sensing. My research is, I'm interested in coastal biogeochemical processes using field, satellite, and modeling approaches. And in my research, I use field and lab optical measurements and satellite ocean color remote sensing to get estimates of phytoplankton biomass, which is important in ecosystem studies, CDOM, which is color dissolved organic matter, the optically active fraction of dissolved organic carbon, which is also very important in carbon cycling, and SPM or suspended particulate matter, which is used very important in restoration studies in Louisiana coast and water quality. My areas of study has been in Louisiana, Texas coast. I've done work in Galveston Bay and Apalachicola Bay in Florida. So as you can see in this imagery, coastal and estuarine waters are optically complex due to the large influx of terrestrial material and high productivity. And therefore, my goal is to develop improved ocean color models for turbid coastal waters and applications, and for example, to assess extreme events. And this is a flow chart that shows some of the algorithms we use to get estimates of POC, SPM. And we applied that to Galveston Bay, where we studied a 500-year flood event in 2017 following Hurricane Harvey. As you can see, river discharge increased from 50 cubic meters to about 4,000 cubic meters. So we used a series of satellite imagery, as you can see here, and we calculated the fluxes of material that came out from the associated with the storm. We also recently we did some work on phytoplankton dynamics, and this is an example following Hurricane Barry in 2019. This is an ongoing project, NASA APSCOR funded project with my colleagues, George, Kanchan, and Victor. And we have done a lot of field work, actually extensive field surveys in 2019, 2018. And as part of that work, we developed bioptical algorithms to retrieve chlorophyll biomass, phytoplankton biomass, and also phytoplankton size structure, which is fundamental for biological studies. As you can see, we have highly dynamic phytoplankton dynamics in this region. We have micro phytoplankton coming up. These are the large phytoplankton like diatoms before the storm, and then pico phytoplankton, which are really the small size. You can see these large changes in this imagery. Anyway, most of this funding has come from NASA, and I would like to thank my previous students, Ishan Joshi, who contributed, and Bingqing Liu. He is a postdoc at Scripps Institute, and Bingqing is a postdoc at the Water Institute of the Gulf. Well, thanks.